really gives me... Hello everyone. For those of you who don't know me, my name's Tracy Dixon. This is my second year, I think, on the MND Tasmania board, and it really gives me great pleasure to be here. I'm a researcher at the University of Tasmania who works on motor neuron disease, and I've worked on this devastating disease for over 20 years. But for members who are joining us or listening to this recording, you may not realise that at the university we actually have a very large group of researchers who work on MND, and we probably would be nationally one of the largest research centres dedicated to MND research, with probably close to 50 people working really hard to understand the causes of MND and to develop new therapeutics. So it gives me great pleasure in introducing one of my collaborators and colleagues, Professor Anna King and Dr. Sharn Perry. They work in a part of the university called the Wicking Dementia Research and Education Centre, and again are also dedicated to researching the causes and developing treatments for MND. So Anna, thank you for joining us today, and Sharn, we're really looking forward to your presentation, and I've over many years enjoyed our collaboration and how particularly nationally and internationally, MND researchers are really known for the collaborative way we work together, and it's something I'm very proud of, and I'm sure you are as well. So welcome, Anna and Sharn. I'll hand over to you, and thank you for joining us today for your presentation.
Well, thank you, Tracy. So I'm just going to try sharing my screen so that, because we've got a bit of a presentation to share with you, we just might have to make sure we're getting the right screen up. I'll just do that now. There we go.
Perfect.
Is that the right display, Tracy, or is that the...
Yes. No, that's it. We can see the full slides and slide number one.
Cool.
Brilliant.
Okay, I can't see anyone now, so I will just talk to the screen. So thank you very much for inviting us to present today. So yes, as Tracy mentioned, and thank you for that introduction, Tracy. I'm Anna King, and I'm here today with Dr. Sharn Perry, who is a work colleague, and we're both from the Wicking Dementia Centre. But as Tracy mentioned, we work alongside a number of other researchers at the University of Tasmania who are all passionate about motor neuron disease research. And I think that team is really growing at the moment. So we thought today we'd talk to you about two of our current projects, but they're really part of our bigger picture of research, which is really to try and find treatment strategies for protecting the motor neurons in motor neuron disease. And in particular, we're focused on not just protecting the motor neurons, but making sure that they maintain their connections with other neurons or with the muscle, because if we protect the neurons, but they're not talking to each other, we still won't maintain that motor function. So I'm going to start off and give a bit of an introduction to our work, and then Sharn's going to talk in a bit more detail about two of the projects that we're working on. And then at the end, it depends whether we've got time or not, there's some more research that we're sort of more heading into that I'll talk to you about, but we might not have time for that, so we'll just see how we go. So before I start, I need to give you teach you a little bit of neuroscience. So up here I have an image of a neuron or a nerve cell, and this nerve cell is growing in a culture dish. So it's been taken out of a rodent brain, and it's been growing in a dish. And it's filled with something that makes it fluoresce. So that's why you can see it as a dark image. But what you can't see in this image is that there's a whole heap of other nerve cells growing in this dish that you can't see that are talking to this nerve cell. So this big round blob here is the cell body, and this is really the control center of the cell. It contains the DNA, but it also makes most of the things that the cell needs and needs to transport these out to the rest of the cell. These big fat processes are called dendrites, and these receive information from other cells, from other nerve cells. And these little bits here are called spines, and these are the actual synapses, the talking points between the nerve cells. But what you can also see in this image is these little fine structures here. And these are called axons, and these are how the neuron communicates with other neurons or with the muscle cells. So if you look at the schematic over here, you can see a nerve cell and the dendrites, and this is the axon here. And this is where the message is sent out to talk to other nerve cells via these terminal points, which connect onto these synapses or onto the muscle at neuromuscular junctions. And when we draw schematics of neurons, we often draw them like this with a big cell body and dendrites and these little axons. But in reality, neurons look more like this. So this is an actual nerve cell in a mouse brain. This is a cross section of a mouse brain. And this pink here is an actual tracing of a nerve cell. Up here, you can see the cell body and the dendrites, but all the rest of these pink lines are the axons. And you can see how big the axon is compared to the cell. And this can make axons very vulnerable. So most of the things that the cell needs to do, the control center makes most of the proteins that are sent out. And you can imagine that it's quite hard for a nerve cell to maintain such big axons. And they need to be able to transport things out to the axons, and they need to be able to communicate with the axon, sending messages backwards and forwards to these axons. And this requires an awful lot of energy. And it makes the neurons quite vulnerable. Just to highlight further how big these axons are, a few years ago, I was invited to present some of our research in China, and I tried to come up with an analogy. So this is the building where we work, and this up on the top floor, the 5th floor, is where our laboratories are, and it's really quite big. But I worked out that if this whole top floor of our laboratory was a nerve cell body, the control center, then the axon could go from Tasmania right the way to China. And so I think this really highlights how big these axons are. And when you think about a neuron, a big neuron, most of the neuron is actually the axon. And this is really important in motor neuron disease because motor neurons have very large axons. So as probably most people know, there are 2 types of motor neurons that are vulnerable in motor neuron disease. We have the upper motor neurons, which are in the brain, and they send very long axons down the spinal cord to innervate the lower motor neurons that send very long axons out to the muscles. And so axons of motor neurons are quite big, and we also know that they're vulnerable. So we can see that they're vulnerable because we can see that they degenerate in the spinal cord and also in the nerves. And we think that they degenerate quite early on in disease, but what we don't know is whether they degenerate first and then drive the degeneration of the nerve cells, or are they just part of the overall degeneration of the nerve cell as a whole. So I mentioned that we were particularly passionate about trying to work out ways of maintaining these axons. And you might ask, why is that important? Surely if we protect the nerve cell, we would also protect the axon, which is just part of the nerve cell. But actually that turns out not to be quite true, because we now know that axons are quite separate in some ways to the nerve cell body, and they can have separate degeneration mechanisms. We know that there are mechanisms that happen in axons that don't happen in the cell body. And this means we might need to protect these axons separately from the cell body. We know that they can trigger their own breakdown in response to injury. And researchers around the world are now trying to work out what some of these degeneration mechanisms are. And we don't know whether there's just one degeneration mechanism or if there are lots, but we're a little behind our knowledge of what's happening in the cell body. And that's some of the work that our group is particularly interested in doing. So one of the mechanisms involves a protein called SARM1. And this was discovered about 10 years ago. And this protein, when it's activated in the axon, initiates the destruction of the axon. And we're not quite sure how this protein is involved or if it is involved in motor neuron disease. But we've become interested in this protein to work out if we prevent the activation of this protein, can this protect axons in disease? So this is some work that we've done in an experimental model, looking at whether if we stop the activation of this protein, can we protect these axons? And what you're looking at here is actually the optic nerve. So this is a nerve that connects the eye to the brain. And nerves contain lots and lots of axons, and this is a cross section. So what you're looking at in this first picture, all these round circles are cross sections of axons in the optic nerve. And you can see this is a healthy optic nerve, and you can see these lovely axons here. When we induce an experimental model of disease, you can see that a lot of these axons are lost. So there's a few still left, but a lot of them have been lost, and the ones that are there are not looking very healthy. In this same model, if we stop the activation of this protein, actually we're removing it, but we stop in the activation of this protein, in this same disease model, we can see the protect. And so we're interested now in investigating this protein in motor neuron disease to work out whether it could be a protective strategy for motor neuron disease to protect these axons. The second mechanism that we're interested in that Sharn's going to talk about today involves what's known as the cytoskeleton. So axons, like all the rest of cells and neurons, contain a lot of structural proteins. These are like a skeleton. They maintain the structure, but they also have a number of functions. And one of the proteins we're particularly interested in is called the microtubules, and that's an image of a microtubule here. These are important because they're important for transporting things around the cell, and axons require a lot of transport from the cell body out to the periphery. They need to transport components that the cell needs, but also energy supplies and also messaging between the axon and the cell body. And we know that these microtubules become broken down when the axon degenerates. And so in some research, the preliminary research we have done, we wanted to find out what are the initial changes that happen to these microtubule proteins before an axon starts to degenerate. And we identified that just before degeneration, there is a change to the microtubules, which is called deacetylation. And you can see this is normal axons, healthy axons, and here you can see they get a decrease in this acetylation. You don't really need to know what that is, but we know that this change happens. And when microtubules become deacetylated, it weakens them. And there are drugs that we can use that will prevent this deacetylation. And so theoretically, this should strengthen the microtubules, and maybe this could help prevent the degeneration of axons. So over here, we have another experimental model. This time it's in cell culture. And you can see these lines here are the axons. These are healthy axons. And this is the disease model where you can see that the axons are all broken down and fragmented. When we add this drug that prevents this deacetylation of the microtubules, prevents this change to the microtubules, you can see that the axons become protected. These are just two different doses of the drug. So we're now wondering whether we can use this drug to try and protect axons in motor neuron disease. So now I'm going to hand over to Sharn Perry, who's going to talk to you a little bit about how we've been testing these treatment strategies in animal models of motor neuron disease.
Just do a little swap. Hello, everyone. Thank you, Anna. I'm going to carry on and talk a little bit more about what we're doing at the moment, but our research questions are really founded in what the field has discovered in the past 10 years, as Anna was saying, but also what our own preliminary research is suggesting. So I'm going to talk about two main projects that really are answering two different questions, but we have the same purpose to see if we can stop the axon from degenerating. So our research investigates whether we can protect the axons from degeneration by the two different mechanisms that Anna has talked about. So one is this spontaneous self-destruction pathway that's really activated by the protein SARM1. And the second is whether stabilizing the microtubules can help to prevent the axon from degenerating. And that is by applying a drug to stop an enzyme that acts on these microtubules. So our first project really focuses around the role of the protein SARM1 and whether SARM1 is involved in axon degeneration in MND. So we kind of hypothesized that when we have a regular SARM1 protein in our neurons and MND disease is occurring, then the active SARM1 protein will cause axon degeneration, or it will trigger axon degeneration in some way. But we're interested in knowing that, if we remove the activity of SARM1, whether this can protect the axon. Our second research question is looking at whether we can stabilize the axons to prevent their axon degeneration. And as we said before, we're looking specifically at the microtubules and really the role of an enzyme called HDAC6. And its role is to modify the microtubules to make them stable or unstable. So we want to apply a drug that blocks the activity of this enzyme with the hope to make the microtubules more stable to strengthen the axon. All of our work that I'm going to talk about focuses on using mice as a model of MND. So we have mice that will develop the symptoms and the pathology, so what the changes that happen in our nervous system of ALS. So we have mice that will develop the symptoms of ALS, and then we look at our two research questions in two different ways. We have mice that are born without the SARM1 protein. And then we have mice that will develop the ALS symptoms, and we give them a drug to stop the activity of HDAC6. So in these two separate experimental cohorts, we are tracking and continually monitoring the animals' progress, their clinical disease progress, and also their motor function throughout their life. And at the end of the experiment, we take their spinal cord and their sciatic nerve. And we do this so that we can have a look at the motor neuron numbers in the spinal cord and whether we see any type of peripheral axon loss. So I'm going to show you results today from the spinal cord and the sciatic nerve in both of our animal models. So our first question was really around whether the protein SARM1 has any role in ALS-driven axon degeneration. And firstly, we were interested in whether we're able to protect the motor neurons. So we took a section of spinal cord and we cut directly through the middle as if you were taking a section straight through the mouse. And we took those sections of spinal cord and we dyed them so that we could see the neurons. And in this picture here, all of these large purple neurons are motor neurons. And we looked at mice that didn't develop ALS, so they're control animals. We looked at mice that developed ALS, but they had an active normal SARM1 protein. And then we looked at mice that developed ALS, but we had removed the activity of this SARM1 protein. So we counted up all of the motor neurons in the different animals and we quantified them. And that's what we're looking at in this graph here. And we saw that in mice that developed ALS, and they are the green bars here, they had a lower number of motor neurons than the control mice. But what we were really interested in was looking at the difference between mice that had an active SARM1 protein and the mice that didn't have an active SARM1 protein. But we didn't see that the mice without an active SARM1 protein, they didn't have more motor neurons. So from this, these results suggest that the loss of SARM1 wasn't very effective in preventing motor neuron loss in this mouse model. So we next looked at the sciatic nerve. So here we've taken the sciatic nerve out of the mouse, we've cut it directly in half, and that's what we're looking at in these images here. Again, we've dyed the nerve, and all of these little white dots are the axons that are running through the sciatic nerve. And we're looking again in mice that don't develop ALS. We looked in mice that do develop ALS and they have an active SARM1 protein. And we looked in mice that do develop ALS, but they have an inactive, or they don't have the active SARM1 protein. And what we found here was that in mice that developed ALS, they had a lower number of axons in their sciatic nerve. So we can see that this green bar, which are the mice that develop ALS, is lower than the control mice that don't develop ALS. But what we were really intrigued to see was that in mice that didn't have the SARM1 protein, they had a higher number of axons. You can see that it's almost back to control levels. Not only that, but in mice that develop ALS, that the axons that they did have running through their sciatic nerve were much smaller. But when we removed SARM1, the size of the axons increased back to control levels. So this was really encouraging to see, because it looks like loss of SARM1 actually does protect peripheral axons in this model. When we looked at our second question, which was whether blocking the activity of an enzyme to stabilize the axon, whether this can help in preventing axon degeneration, we did exactly the same thing. So you might see that these pictures look similar. And this is part of a really large study. So at the moment, we're looking at the role of blocking this enzyme in one mouse model, but we've got a lot of other mouse models where we're testing this drug in them as well. So I'm just showing you preliminary data from one mouse model, but it is quite a large study, and we've still got a lot of work to do to see the effects of this drug in other mouse models, as they don't always have exactly the same clinical symptom presentation or trajectory of disease. So again, we took a section of the spinal cord and we dyed the spinal cord to see the motor neurons, these purple cells, and we counted the cells in mice that don't develop ALS, in mice that do develop ALS, and then in mice that develop ALS, and we gave them the drug. And you can see here, these bars are very similar between the ALS mice and the mice that develop ALS, and we gave them the drug. So it looks that blocking this enzyme to stabilize the microtubules wasn't effective in protecting the motor neurons. So then we wanted to look to see whether we could protect axons. So again, we've got a sciatic nerve that we cut through, and we've stained all of the axons, or we've dyed all of the axons in the sciatic nerve. And this time we dyed all of the axons red, but we dyed the motor neuron axons in green. But we were really just interested in looking at the total number of axons in the nerve. So we only counted the red axons. And here we're looking at the number of axons between mice that developed ALS and mice that developed ALS, and we gave them the drug. And again, we didn't really see that blocking the effect of this enzyme was effective in protecting axons. So at the moment in this mouse model, we can't really say that blocking the effect of the drug is effective in protecting axons or the motor neurons. But just because we didn't see an effect of this drug in this model doesn't mean that the drug isn't effective. As I said previously, we are looking at the effective blocking of this drug in a lot of other animal models, and that work is ongoing. But also our collaborators are seeing protective effects of this drug in other mouse and cellular models. So we are continuing our investigations to see what else we can find. So with that, I'll hand it back to you, Anna. Yeah.
Cool, thank you, Sharn. I think we probably haven't got time to talk about the new areas we're going to, so maybe you can invite me back another time to talk about those. But I suppose just to sum up, what we think our data is showing is that we are finding some protective effects of the axons, but obviously not of the neurons themselves. And so this might mean that we need to combine therapies that protect axons with therapies that protect the whole motor neuron as well in some kind of combination therapies. And hopefully we'll get the opportunity to look at some of these things in the future. I just skipped through these slides, but I'd just like to end up by a number of acknowledgements. I have a list of some of our team members here. I'm not going to go through all of them individually, but here's a bit of a picture of our group celebrating a paper, I think, that was celebrating a successful paper submission. And I'd also like to thank all our funding bodies, particularly Fight MND and MND Australia, MND Research Australia, for funding over a number of years. It's been really amazing to have. Thank you. Just work out how to stop sharing. That's good.
That was fantastic. Thank you, Anna and Sharn.
Has anyone got any questions?
I don't have a question, but I guess just to make a comment, Anna, that people may have heard Anna finish with this idea of combination therapies, and it's really how we're seeing the MND research field evolve, recognising, and you may remember from Brad Turner when he spoke to us last year, talking about a more personalised medicine approach for MND people, understanding that MND is a very complex disease and that different types of MND there may be many different types of MND that will require a different therapeutic approach. So it's very exciting when you see preliminary data that suggests that, going after the axon is a good target, and potentially using these drug approaches in combination could be a successful way to go. Thank you, Anna and Sharn.
I'll just follow up from that, Tracy. So some of the work that I didn't talk about that we're starting up a bit more is, as Tracy was talking about, this more personalised approach. We're hoping to develop new what's called biomarkers. So when we're dealing with mice, it's very easy to understand what's happening to the nerve cells. But when we're dealing with people, we can't understand what's happening to the nervous system. It's very difficult to know what's happening to the nerve cells. And so what we use is what we call biomarkers, so biological markers that are indicators of what changes. And so some of the new work that we're doing is trying to develop better biomarkers that can tell us more about where in the nervous system degeneration is happening, what parts of the nerve cell are degenerating. So we hope to be able to present some of that work later in the future. And we've got a few collaborations across Australia doing that. So. Oh.
I've got a question for Anna. Do you know how ridusol works? What's the connection?
Yeah, so ridusol is a drug that we think prevents what's called hyperexcitability. So that's to do with the firing of the nerve cells. And so we know in motor neuron disease that the nerve cells become hyperexcitable. They fire too much. And this might be one of the causes of them degenerating, because it can increase sort of dangerous chemicals within the cell. And some of these models that I showed you in our preliminary data was actually a model of that hyperexcitability, because we have shown that hyperexcitability can cause the axons to degenerate as well. So ridusol is protecting that pathway, but what we understand now is that there are many other pathways. Potentially, as you get later on in the disease, there are many other pathways that have been activated, and so we might need to provide additional drugs to help stop some of these other pathways that have become activated in disease.
Thanks.
I have a very silly question, perhaps, if you'll indulge me. My understanding was that part of the challenge with MND is that we don't actually know what causes it. So we know what happens when someone has it, but we don't know what causes it. So with that in mind, how is it that you're able to introduce it to mice? Like, how do you end up being able to practice on mice that have ALS if we don't know exactly how someone can get it?
That is not a stupid question at all. That is an excellent question.
Okay, thank you.
So, you know, we use, most of our mouse models are developed on, you know, there's, as you probably know, there's some types of motor neuron disease and ALS that are familial. They're due to a gene mutation. So we use these gene mutations to develop these models. They're still not exact models. They are just models, and they're all the animal models that we use are very different. But we can induce them to have the same sort of pathological features that we might see in human ALS. And we have to be very careful in how we interpret the data that we use from animal models, because they are not animals with motor neuron disease. They are animals that are modeling it. So for our work, for example, we're looking at whether we can protect the axons. And we can't definitively say if we protect the axons in an animal model, whether that is going to be the same in humans, but you know, we do need to start somewhere, and that's why we need to use several different models to make sure that, you know, it's across a number of different things, and also that we're not missing things. So we use these familial models, or we can drive changes that we know occur in disease. But you're absolutely right, we still need some basic understandings of motor neuron disease and how it's different in different people, because that's another thing we're beginning to understand, is that, you know, not everyone is the same, and everyone can get this disease in different ways and potentially through different causes. And, you know, going back to some of these basics is incredibly important. But most of our strategies are sort of looking more at the protecting that more downstream, the common things that might happen for most people, this actual neuroprotection, whereas other strategies might be trying to look further upstream at more individual cases. So yeah, that's a really good question.
It's fascinating work that you do. Thank you for taking the time to meet with us today.
Thank you for inviting us along.
Well, I've got another question, Hannah.
Okay, great.
When my symptoms first started to emerge, gosh, was that 3 years ago, it was really the left leg. And at the time, you know, it felt like that I had a back injury, but I knew I didn't have a back injury. It was a sort of a lower back, you could say, ache on the left-hand side. And anyway, the left side leg has always been lazier than the right side leg. So it's really been around for a long time. But the surprising thing is why it would also then, you know, the condition affects the swallowing and the breathing, because the upper body otherwise seems to be fine. So why is it so selective, and why does it affect the swallowing? Why is that you could say a characteristic of MND?
Yeah, so that's really interesting that you think that the symptoms seem to have been for a long time. And what you're talking about is one of the things we really don't understand yet, why it spreads from one region to another. We don't even understand whether, you know, it's driven from the changes in your upper motor neurons in your brain, or whether it's driven from your lower motor neurons in your spinal cord, or is a combination of both. And it's one of the really important questions about whether the changes can spread from cell to cell, or whether it's just that you know, something is happening and there's vulnerable cells that are starting to generate at different times. And it's one of the important questions that we really don't know yet. But we do know that there are some motor neurons that are less vulnerable, and they're the ones that control your eye movement, and also the ones in the ONUS nucleus that control your bladder. And we know that these are less vulnerable, and there's a lot of research going on trying to work out why some motor neurons are more vulnerable to others, but also that spread through the spinal cord, why some areas become affected and not, but it's not something we know yet.
Thanks.
I think also, Greg, what you've described is that real variability. And when drug trials were initially being proposed for people with MND, all people with MND were sort of lumped together for a trial. Whereas now we're trying to do a better job at recognising that heterogeneity and trialling drugs in subpopulations. So trialling drugs in people who have an onset pattern similar to yours, you know, starting in the lower limbs and then progressing to bulbar, but in the hope that maybe a drug might work for those types of MND, but it might not work for the people who start the opposite way, start with bulbar presentation. So, but when you mix everyone together, you sometimes lose that drug effect. You might not see when it's working for some people. So really understanding those patterns of presentation and progression and organising the drug trials to match that and to sort of understand those patient subgroups is a really important sort of progress in the field. And we hope that it will mean that we might detect positive effects more easily by recognising those differences.
Any other questions for Anna and Sharn?
Yeah, I've got a question. So it would seem to me that from what I've heard, it takes a long time to diagnose MND, and people go through a lot of physicians and to diagnose it. It seems to me that the sooner you could get somebody onto a drug that may be effective, that might be more effective. Is there a lot of work being done on diagnosing MND earlier?
Yeah, there is, Steve. And so some of the work I was going to present actually is looking at these biomarkers, because, you know, there's the very early stages of MND when changes are starting to occur, but there's no clinical symptoms at all. And we don't have any biomarkers at that stage that could be indicating that changes occur. But then there's the next stage where, you know, people might be getting some very subtle symptoms, and we might not yet know that that is MND, or it might not even be suspected, but people might be noticing some very small changes occasionally. And we have a biomarker called neurofilament light, which is released from degenerating nerve cells. And this is thought to be going on many, many years before, well, not many years, but a few years before clinical symptoms actually, you know, we can actually start a diagnosis. And if we could pick up this early neurodegeneration from people having sort of more minor changes that they're just a bit aware of, we might be able to make an earlier diagnosis. And these proteins are released from these cells and they get into the CSF, but they also get into the blood, and they're only present in very, very small concentrations. And until recently, we haven't been able to measure them in the blood. But obviously, if you can give a blood test, it's much, you know, you could give a blood test to virtually anyone who comes to the doctors, because it's, you know, it's not that invasive. And if we can give this blood test to determine whether there is neurodegeneration occurring, it might help with an earlier diagnosis. It's not specific for motor neuron disease, but at least we'd be able to see that there was something going on that could then warrant further investigation at this earlier stage.
Perfect, thank you.
Well, if there are no other questions, I guess I'd like to extend a thank you on behalf of the board to Anna and Sharn for coming along and presenting. I think we all thoroughly enjoyed that, and a lot of it goes over my head, but the bits I can catch on to, just think it's such important work that you're doing, and we all appreciate the effort that you and your team are putting towards this. So thank you.
Thank you very much for inviting us.
Yeah,
Lovely to meet everybody.
Yeah, thanks.
Should we go now, or should we?
Oh no, well, I was going to say that probably closes the formal proceedings for the day. So if people would like to stay on the line and have a cuppa and a chat, then you're most welcome. But if people have other things to go off and do, then you'll be forgiven for leaving the meeting as well. So yeah, thanks everyone for coming along.
I'll stop recording now if everyone is happy with that.
Sure. Thanks, Lucy.